Imagine a lab where the experiments don't just run themselves, but they actually learn and adjust on the fly,
Yeah.
like a super smart robot scientist. Today, we're taking a deep dive into something truly, well, revolutionary: autonomous perfusion systems. We've gathered your sources on this cutting-edge field articles, research, the works, and our mission is to break down how machine learning isn't just assisting, but fundamentally changing biological research and manufacturing.
Yeah, and these aren't just like simple automated systems. They're really intelligent platforms. They seamlessly integrate sensors, pumps, valves, and some pretty sophisticated machine learning algorithms all in a closed loop.
A closed loop,
Yeah.
meaning it feeds back on itself.
Exactly. The goal is to meticulously maintain optimal conditions for, well, all sorts of things: delicate cell cultures, engineered tissues, even those complex organs on chips.
Okay, so by the end of this deep dive, you listening should have a really clear understanding of how these systems work, where they're being used right now, and why they're such a potential game changer for science. And importantly, without getting totally bogged down in jargon. Okay, let's get into it. So we've set the stage with this vision of a robot scientist, but we can start with the basics. At its core, what are we actually talking about when we say autonomous perfusion?
Right. At its heart, it's a closed-loop system. So data is continuously collected, it's analyzed by machine learning, and then it's immediately acted upon to control these biological environments.
Like a smart feedback loop.
Exactly, a highly intelligent one. This means the system is dynamically adjusting parameters, things like flow rates, nutrient feeds, gas tensions, all in real time.
So it's not just following a preset script, it's actually learning as it goes. That seems like the key difference, isn't it?
Yeah, precisely. It responds to live incoming data. It's constantly adapting to changes, maybe in the environment or in the biological sample itself.
Yeah.
And this level of automation, well, it significantly cuts down the need for manual human intervention.
Right, less fiddling around in the lab.
Less fiddling, yeah. And it can self-correct issues like fouling, that unwanted buildup in channels.
Ah, okay.
And it's brilliant at optimizing multiple variables at the same time, something that's incredibly hard for a person to do manually.
Okay, this really does sound like something from a sci-fi lab.
Oh, yeah.
So for everyone listening, where are we actually seeing these systems in practice today? Is it still mostly research or?
Oh, no, we're seeing widespread application already,
Yeah.
mainly across bioprocessing and also in engineered tissues.
Mm.
So in bioreactors, for instance, closed-loop machine learning is being used to precisely regulate nutrient feeds, environmental parameters for cell cultures.
Yeah.
There's one quite impressive example from your sources: a 24-parallel perfusion bioreactor platform used for monoclonal antibody production, MABs. And the whole thing was operated by an autonomous software agent.
24 at once.
Mm.
Wow. And it's improving itself. How smart is this improvement? Is it just tweaking or finding new ways?
It's genuinely optimizing. This particular system, it combined Bayesian experimental design, which is basically a smart way to decide what experiment to run next for the most info, plus a digital twin model and ML feedback. In over a 27-day run, it autonomously increased volumetric productivity and kept cell viability high. So yeah, actively optimizing.
That's amazing.
And there's another example, Traylor et al. They showed in silico, so in simulation, that a reinforcement learning agent could maintain a two-species microbial co-culture at target levels. And it actually outperformed a traditional PI controller, which is like a standard, simpler feedback system.
Right, the old thermostat kind of approach?
Sort of, yeah. And the ML did better, especially when sampling wasn't very frequent. It really shows ML can handle complex long-term automation.
Okay, so bioreactors. What else?
Well, beyond that, tissue engineering. Perfusion bioreactors for scaffolds or organoids, they also benefit hugely from ML control for, say, dynamic oxygen and nutrient supply. And then there are the organ-on-chip systems, OOCs. These microfluidic devices, they often need incredibly precise flow control. So here, machine learning, especially reinforcement learning, can dynamically adjust pressure or flow actuators. It compensates for tiny differences between devices and maintains physiological shear, that's the fluid force on the cells.
Okay, keeping it realistic for the cells.
Exactly. For example, RL-based deep Q networks, they've used image feedback to stabilize microfluidic flows over long experiments. Your sources even mention multi-organ OOC platforms using robotic liquid handlers to link and perfuse up to 10 chips in parallel.
But chips at once. That's a massive leap in throughput, surely. What strikes me here is it's not just about keeping things stable, right?
Yeah.
It's enabling really complex stuff, like dynamic multi-drug screening.
Precisely. And that's thanks to having these on-chip optical and biochemical sensors constantly feeding data back to the ML controllers. So yeah, in summary, ML-enabled perfusion control is popping up everywhere. High-throughput small bioreactors, patient-specific tumor-on-chip models, interconnected organ chips. It offers automated operation across a really diverse range of applications.
Okay, we've seen where they're used. The intelligence behind them is obviously the machine learning.
Yeah.
So let's dig into the how. What specific kinds of AI are actually running the show here?
Right, so different ML approaches have been explored. Reinforcement learning, or RL, that's a big one. RL agents, they learn policies, essentially, strategies for adjusting control inputs like flow rates or nutrient feeds based on what they observe, the state.
Right.
Techniques like DQ networks, DQNs, Q-learning, neural-fitted Q-learning, they've all been applied successfully. Dressler et al., they trained both DQN and model-free episodic controllers to manage things like laminar flow and droplet size in microfluidics, just using image feedback. They actually called the stability superhuman.
Superhuman stability, wow.
And Trelar et al. They used a neural-fitted Q, or NQL, agent to control two different nutrient feeds in a continuous bioreactor, keeping two microbial populations stable. And the policy it learned was this bang-bang type, which beat a standard PID controller when sampling was sparse.
Bang controls, that sounds pretty abrupt. So am I getting this right? RL is like teaching the system to figure out the best moves by trial and error. And bang, because the control is just full on or full off, no messing about.
Exactly right. It's decisive. And that learned action is often surprisingly effective, especially when the system is learning through trial and error to hit a specific goal. RL is really attractive because it's often model-free, meaning it doesn't need a perfect mathematical model of the biology beforehand.
Okay, that makes sense. It learns the rules itself.
Yeah. And it can optimize for somewhat arbitrary rewards, like just maximizing productivity. The main challenge, though, is it often needs a lot of exploration, lots of trial runs, which can take time.
Got it. What else is in the AI toolbox?
Well, then you have supervised learning and deep learning. These tend to be used more for the sensing and prediction side rather than direct control. So, for example, convolutional neural networks. CNNs, which are great with images, they've been used to analyze images from the chips.
Right, like recognizing patterns.
Exactly. Liu et al. They trained a CNN to automatically classify vascular network morphology in OACs. That really helps enable high-throughput screening. And supervised regression models, things like artificial neural networks, ANNs, or random forests, they can predict process variables, maybe cell density or metabolite levels based on sensor data. And those predictions can then feed into other controllers. And finally, there's Gaussian process, or GP, and Bayesian methods. These give you probabilistic predictive models. They don't just predict, they give a sense of uncertainty, too. Mueller et al., for instance, developed a hybrid model using stepwise GP regression. It was trained on real experimental data and coupled with Bayesian optimization to help design the experiments in those bioreactors. This GP model acted like a digital twin. It learned the bioreactor dynamics, cell growth, viability, and used that to guide the feeding strategies.
A digital twin of a bioreactor. That's amazing. It's like having a virtual simulation that perfectly matches the real thing, predicting how the cells will behave.
Pretty much, yeah. It serves as this predictive model for estimating the state and recommending control actions. And this approach, it allowed them to do transfer learning between different cell lines and achieve really robust optimization over multi-week cultures.
So it's not just predicting. It's like running virtual experiments first.
Exactly. It lets researchers test countless what-if scenarios, virtually different nutrient strategies, environmental tweaks before committing to a real, often expensive and long physical experiment. It massively speeds things up. But in practice, many cutting-edge systems actually combine ML with conventional control. They might use ML to smartly inform or tune the set points for existing controllers like PID or MPC.
Okay, so it's layers of intelligence working together.
Yeah.
Now, the ML is the brain, clearly. But for that brain to work, it needs incredibly good eyes and hands, the sensors and hardware. What are the key bits of kit making this real-time action possible?
You're spot on. The hardware is critical. They need diverse sensors and actuators. Common elements are various types of pumps, peristaltic, syringe, pneumatic, plus valves for really precise fluid flow control, and often robotic liquid handlers for complex tasks like linking multiple organ-on-chip.
Like the 10-chip system you mentioned.
Yeah, exactly. There was an automated platform called the Interrogator that used 3-axis robotics, peristaltic pumps, and even an onboard microscope to perfuse an image up to 10 organ chips continuously.
Wow.
So data collection relies heavily on First, optical imaging. High-res cameras, bright field, phase contrast, fluorescence give rich data on cell shapes, markers. Time-lapse microscopy can feed into those CNNs we talked about for automated cell counting or classifying cell types. And in microfluidics, you usually have imaging windows for direct observation of the flow.
Okay, so seeing is believing, literally.
Definitely. Then you have electrochemical and biochemical sensors. Miniaturized sensors measuring chemical parameters in situ right there in the culture. Examples. Dissolved oxygen probes, pH electrodes, electrochemical sensors for glucose or lactate, often built right into the chips themselves.
Built in, that's neat.
It is. And Trujillo de Santiago et al., they did something really clever. They used a commercial continuous glucose monitor, like the Abbott Freestyle Libre people use for diabetes.
Wait, the actual thing people wear?
Yeah. They clipped it onto a tumor on chip to measure glucose in real time. Such a smart integration of existing accessible tech. You also see optical oxygen sensors, or optodes, and pH-sensitive dyes polymerized right into the channels. And multiplexed biosensors can detect several biomarkers or metabolites at once, giving a richer picture of cell activity.
Okay, so chemical sensing is key. What else?
Then there are the physical sensors. Flow and pressure sensors are vital for ensuring the perfusion rates are accurate. Microfabricated flow meters, pressure transducers track pump performance very precisely. You can also have microelectrode arrays, MEAs, recording electrical signals in heart or nerve cell cultures.
Ah, for function, not just chemistry.
Right. And impedance sensors measure cell barrier integrity, things like tear, transepithelial, electrical resistance. It tells you how tightly cells are connected, which is crucial for tissue health. All these sensor outputs, they get digitized and streamed in real time. As Q et al. Noted, integrating a variety of online physical and biological electrochemical sensors enables monitoring of nutrients, metabolites, shear forces, pH, and oxygen. And all this high-dimensional data is then the fuel for the ML algorithms to drive the feedback loop.
Okay, so we've got the smart brain, the ML, and the sophisticated eyes and hand, the sensors and hardware. How does it all actually work together? What's the sort of the rhythm, the heartbeat of this autonomy?
Right, this continuous closed-loop feedback control, that's absolutely the core concept. Sensors constantly monitor the culture. The ML processes that data almost instantly, and then actuators apply the necessary corrections. It's this ongoing cycle.
So like measure, think, act, repeat.
Exactly, a typical cycle, sample sensor data, and for the current state, using the ML model, compute the desired action, physically adjust the hardware pump speed, gas mix, temperature, whatever it is. So for instance, in a bioreactor, the controller might use an ML model to estimate live cell density, maybe from optical measurements, then instantly adjust the perfusion rate to maintain a target growth rate.
Okay, now here's where it gets really interesting for me. How fast does this feedback actually happen? Are we talking like minute-by-minute adjustments or faster?
Oh, it can be incredibly fast, depending on the need. Reinforcement learning is a great example here. Once trained, the agent maps observed states to optimal actions almost instantaneously. Dressler et al., they showed an RL agent adjusting microfluidic inflows frame by frame. That's milliseconds, just to maintain laminar flow stability or hit a target droplet size.
Milliseconds, wow.
Yeah. And similarly, Trailer et al. Had their RL agent check culture measurements every few minutes and decide on that on-off bang-bang dosing for 2 nutrients. And that rapid feedback kept both microbial species really close to their target levels, very accurately.
Okay, so RL is super quick. What about other methods?
Well, closed-loop model predictive control, MPC, is another sophisticated strategy. Here, you have an ML-derived predictive model, maybe a Gaussian process or a neural state space model forecasting future states under different possible control actions. Then an optimizer chooses the sequence of actions that's predicted to minimize the error against your targets. Muller et al., they effectively embedded that digital twin idea right into their control loop. They used GP models to predict cell viability and then Bayesian optimization to choose the feed rates that would maximize the final product yield.
So it's not just reacting to the present, it's actively predicting and optimizing the future of the experiment. That's incredibly sophisticated.
It really is. It truly relies on, as the sources say, all these real-time and continuously collected data combined with AI-based data processing for studying and optimizing closed-loop feedback-based experimental parameters. It's the whole package working together.
This all sounds, frankly, amazing for the future of research and manufacturing.
Yeah.
What are the really big advantages these systems offer right now?
The benefits are pretty significant, yeah. First, vastly improved consistency, efficiency, and adaptability. Automated control can run 24/7 without human fatigue or, error creeping in. That enables much longer, higher throughput experiments than you could do manually.
Right, consistency is huge in biology.
Absolutely. Second, machine learning excels at handling complex, nonlinear, multi-factor dynamics, stuff that's incredibly hard for humans to tune manually. And this leads to, as Dressler et al. Reported, superhuman reproducibility with RL control in microfluidics.
Superhuman, meaning it's just that much more consistent than a person could ever be.
Exactly. Imagine trying to keep flow perfect in a tiny channel for days. Human hands just can't avoid tiny errors or variations. These systems make these minute, continuous adjustments with a precision that can really cut down batch-to-batch variation. Much more reliable results.
Okay, what else?
Well, beyond that, the closed-loop ML can actively compensate for disturbances, things like channel fouling or maybe unexpected changes in cell metabolism. It dynamically adjusts inputs to maintain stable conditions over days, even weeks. And finally, they enable multi-objective optimization. Agents can be trained to, say, maximize both cell yield and viability simultaneously. In one example, an autonomous system successfully balanced volumetric productivity and viability to meet really stringent goals. That's a major achievement.
Okay, that is a compelling list. But,
Yeah.
I mean, with tech this advanced, there have got to be challenges, right? What are the limitations or hurdles people are facing?
Oh, absolutely. These are complex systems to design and validate, no question. One major one is data requirements. Training ML controllers, especially RL, often needs a lot of data or simulation time. This sample inefficiency can mean many experiments are needed, although running things in parallel helps.
So getting enough training data is tough.
It can be. Then there are robustness concerns. There's always a risk of the model overfitting the training data or mislearning if the sensor data is noisy or incomplete. Safety and robustness are critical. You wouldn't want an ML agent suggesting some really aggressive action that pushes things past safe limits unless you have strict constraints built in.
Right, checks and balances are needed.
Definitely. And the hardware integration itself, it's not trivial. Making those on-chip sensors like microelectrodes can be expensive, or the sensors might be fragile. Computational latency is another thing that real-time feedback needs: fast calculations, fast inference, which can limit how complex your ML model can be. And then there's interpretability. Understanding why a complex ML model made a certain decision can be really challenging. That can make regulatory approval tricky, especially in biomanufacturing. And finally, scalability. Many of the really cool academic demos are, in simulation or at lab scale. Translating these successfully to huge industrial bioreactors, that can face significant scale-up issues.
So the promise is huge, but there's still quite a bit of intricate engineering and validation work involved in perfecting them. Given all that, how do researchers actually measure success? How do they know if one of these systems is working well?
Yeah, good question. They assess them mainly by how well they hit their predefined objectives. Common metrics would be things like deviation from a target value, maybe the root mean square error, RMSE, of cell population versus the set point, or process yields like the amount of product made or the viable cell density. Control stability, often measured by variance, is important too. And for reinforcement learning specifically, they track the cumulative reward the agent gets or how fast the learning curve improves. For instance, Triller et al. Show their RL agent learned an effective policy pretty quickly within a single 24-hour run, which is considered fast for RL.
Okay, that makes sense. Let's shift focus a bit to some of the really exciting recent developments, especially stuff from, say, 2020 onwards, that really points to where this field is headed right now.
Yeah, definitely. AI-driven perfusion control has really moved from being mostly theoretical to experimentally proven in recent years. Dressler et al., even back in 2018, they were pioneers, showing deep RL working on real microfluidic devices. Achieving that robust flow and droplet control with image feedback really opened the door. Then Triller et al. In 2020, with that in silico chemostat work, showed RL could maintain two species together, beating standard control, especially with sparse sampling. And crucially, they showed usable policies could be learned relatively quickly, 24 hours on 5 reactors.
Okay, so progress was building. What about very recently?
Well, Miller et al. In 2024, that work is really significant. They presented that autonomous experimental machine for MAB bioreactors. What was revolutionary wasn't just the 24-reactor setup, but how the system integrated prior biological knowledge with on-the-fly learning using those hybrid GP models.
Right, the digital twin idea.
Exactly. It allowed the system to run for almost a month, 27 days, with minimal human input, proactively optimizing feeding to boost antibody yield and keep cells happy. That's beyond just automation. It's like self-optimizing discovery.
That's incredible.
Also, from 2024, Gerzano Flores et al., they showed that clever integration of a commercial glucose meter for organ chips, making real-time metabolic monitoring more accessible, more cost-effective. And Q et al. In 2023, they published a perspective looking at sensor integration and AI for organ-on-chips, really outlining the path towards truly intelligent OSCs by combining continuous monitoring with ML control.
So hearing about these breakthroughs, what does the near future look like? Where is this heading next?
Well, looking ahead, I think key advances will definitely include integrating even richer sensor suites. Think multi-parameter biosensors, more sophisticated optical assays happening right on the chip, and also more advanced machine learning. Things like deep RL, but with built-in safety layers, maybe federated learning where different systems can share knowledge without sharing raw data. We can also expect performance to get even better with physics-informed models ML that incorporate some known physical laws and better transfer learning, so knowledge gained in one experiment can be applied to a new one more easily.
It really sounds like this convergence, this synergy of microfluidics, robotics, sensors, and intelligent algorithms is poised to massively accelerate bioprocess development and organ-on-chip research. Really push the boundaries.
Wow.
What an incredible deep dive that was. From superhuman control of tiny fluid flows to fully autonomous systems making life-saving antibodies and even borrowing everyday health tech to monitor miniature organs. We've really seen how machine learning isn't just helping out, it's actively driving the next wave in biological research and manufacturing.
Yeah, and if you connect this to the bigger picture, the ability of these systems to learn, adapt in real time, handle all these complex biological variables, it truly promises to make biological processes more consistent, efficient, and adaptable than we've ever seen before. Which, raises a really important question. Think about how these autonomous systems, by cutting down on manual work and speeding up discovery, might not only transform how we develop new drugs and therapies potentially much faster, but also how they might fundamentally change our very understanding of human biology and disease models. I mean, what new scientific frontiers could possibly be unlocked when our experiments can genuinely start to think for themselves?
A fascinating thought to end on. Thank you for joining us on this really insightful deep dive. We hope you listening feel much more informed about the world of autonomous perfusion systems. Keep exploring, keep learning, and we'll catch you on the next deep dive.